Welcome everyone. My name is Cora Vacher. I'm the Associate Director for Human Genetics in the Strategic Marketing team. I'm delighted to welcome you all today to this online session. In the next 30 minutes, myself and my colleague Jawar Swaminathan, Director of Informatics in the Global Service and Support team, will present our new end-to-end workflow for 24-hour whole genome sequencing, providing fast answers and the most complete view of the genome. Then, Mark Drost, laboratory specialist at Erasmus Medical Center in the Netherlands, will focus on showcasing rapid genetic testing and the diagnostic implementation of innovative technology in the laboratory. After all the talks are over, we will have a live Q&A session. So please add your question to the speaker today to the Q&A tab. You can also upvote the question you would most likely answer, and we will try to get through as many as we can. This knowledge exchange is being recorded and will be made available on demand after the broadcast. According to the World Health Organization, there are about 132 million global births per year. Out of those, about 106 million happen in hospital or clinical settings. And about 10% of those lead to admission in neonatal intensive care setting. That's about 10 million babies. Out of those, about 4.2 million lead to admission in the highest level of NICU, level 3 and level 4. There are very high level of morbidity and mortality seen in critically ill infants admitted in intensive care settings. So it's a massive global health challenge. And the main reason for NICU admission are respiratory disease, metabolic status, and fever. And genetic might not come to mind first. Take respiratory distress, for example, it could be an infection, it could be prematurity, but it also be the consequences of a number of inherited disorder. I'm not going to go through the list. If you look at hypoglycemia, similarly, it could be due to prematurity, it could be due to perinatal stress, it could also be genetic congenital hyperinsulinism or Beckwith-Wiedemann syndrome. And lastly, fever could come from an infection, could come from perinatal trauma, or can be a response of neonatal Graves' disease. So it's been shown that monogenic disorder are the leading cause, or one of the leading causes of ICU admission and mortality. And there are about 10,000 currently known single locus, or Mendelian genetic disease. Collectively, they really lead to increasing of this admission. And Moore et al. Have shown that, I've published that rapid whole genome sequencing as a first-line test is likely to have a significant reduction effect on the cost of acute care in a private payer system. So in this slide, I wanted to show the diagnostic contribution of the different type of variant in rare disease for the diagnostic. So if you look at small coding variant like SNVs and Indel, they are responsible for about 50 to 60% of the cause of the rare disease. When you start adding large structural variant, copy number, repetitive variant, they contribute to another between 25 and 40%. Non-coding and mitochondrial add between 10 and 15%. And methylation in imprinting disorder, 1 to 2%. So as you can see, there's still about 20 to 30% gap. We all think and hope that methylation will fill some of that gap. I also wanted to show the diagnostic contribution of the most routinely used method. So if you look at chromosomal microarray, in rare disease, depending on the presentation on the rare disease, you can get between 10 and 20% diagnostic yield. With genome-wide whole exome and whole genome arrays, so with whole exome, you get between 25 and 40% diagnostic yield, in particular if you use it in combination with microarray. And for whole genome sequencing in short read, at the moment, the diagnostic yield kinds of plateau at 50% in the best cases. And so Oxford Nanopore technology is really a new generation of molecular sensing technology. But what does it mean? So it means that we can perform real-time analysis that really leads to really rapid insight, but keeping a very fully scalable format. Our technology can analyze both native DNA and native RNA and sequence any lengths, both short and ultra-long read. So we really have any read length technology. All of this is enabled by a tiny pore, nanopore, embedded in a membrane. The DNA or the RNA passes through this pore and it emits a unique electrical signal that we can read. The turquoise line that you can see on the video is the squiggle. It's an electrical pulse. Each of the bases are slightly different. So a T versus an A versus a G emits a different signal. And very importantly, a methylated C emits a different signal than a non-methylated C. And all of that is really interesting, but it wouldn't be very useful if we didn't have a way of analyzing the data. So very importantly, the data analysis itself is fully supported and very intuitive. So AP2Me is Oxford Nanopore desktop application for nanopore data analysis, and it's a very intuitive interface. It provides a range of pre-configured workflow, including a very comprehensive one focusing on human genome variation. Again, it provides a very intuitive and interactive report and also standard output file, including variant annotation and methylation analysis. It can be run locally or on the cloud, and for some workflow, some workflow, including this one, are compatible with real-time analysis. And the same workflow are accessible through the graphical interface, but they can also be run on command line. So if you have collaboration, you can really foster this collaboration between scientists and bioinformaticians, and it makes it very easy to integrate with your other workflow and also your tertiary analysis systems. So Jawar is going to go into much more detail about our workflow for human variation, step by step. But I just wanted to give you all a very high level of what it does. So the workflow enables automated alignment and variant calling. It really enables a flexible analysis. So you can choose from a selection of the type of variant you want to call. In that case, we recommend all of them. The input is a very standard BAM file. And in the result, you'll get SNV, SVs, so structural variant, copy number variant, short tandem repeat, methylation, annotation of the variant, and haplotype phasing, which is very important, especially for methylation in the future. And the output or VCF and BET methyl file, you can also get HTML report. So while the most routine use method in clinical genetics leaves gap, and I've shown you what CMA can achieve, what short-read exome and short-read genome achieve. But with our technology, you really get all the analysis of all the variant in one go. And by all the variant, I mean single nucleotide variant, insertion and deletion, obviously, complex structural variant and copy number, but also deciphering gene and pseudogenes, understanding repeat expansion, especially their size, but are they interrupted or not interrupted? You can look at mosaicism. You can look at mitochondrial variant. That's something that comes up on a regular basis. Yes, you can do that. And very importantly, and I think that's a very hallmark of Oxford Nanopore sequencing, is you get methylation at the haplotype level. So with that, I'm just going to go quickly over the wet lab side of the 20-hour whole genome workflow. So we work with experts in the field to really define what would be a realistic volume input, and we optimize the DNA extraction tab. The workflow starts with DNA extraction using the QIAGEN PureGene kit from 500 microliter of fresh blood. We optimize the DNA extraction further by reducing the elution time, and so that really helps us achieving that 24-hour answer. We do megaruptor shearing, then LSK library prep, which also we have adapted to make the protocol more rapid. And again, I'll go over that in a couple of slides. The sequencing is done on the Promethean 24. We put one sample of a 3 flow cell, and we use ACT-based calling. Again, don't worry about if you don't understand all of that. Jawara is going to go into detail around that. And then we merge the flow cell and perform analysis using the epitome human variation workflow. Variant prioritization and classification can be done with a number of tertiary analysis partners. We have automated compatible output for Fabric Genomics and for Genix. And then those guys can then do report generation and sign out if you need to. So it's a very high-level summary of the spec. We've optimized the workflow to obtain an N50 about 30 kb with an expected coverage of 30x depth for whole genome achieved between 13 and 16 hours. We are aware that some of you might want to go for higher coverage. You can expect to do 40x coverage between 18 and 26 hours of sequencing. Again, we put one sample of a 3 Promethean flow cell, and the tertiary analysis with the compatible partner should take about 2 hours. So again, it takes about 2 hours for the library prep, for the sample and library prep and loading, between 13 and 26 hours for the sequencing, and between 3 and 4 hours for the analysis. So let's go more into detail about the library prep and the sample prep. So ONT prep is very quick and straightforward. And in our case there, it takes just 2 hours plus an additional about 10 minutes for the loading. And we really want to emphasize the fact that, put the emphasis on the fact that it's very user-friendly and it's really efficient. So as you can see on the slide, for the 24-hour whole genome compared to our standard whole genome sequencing, we've optimized the time by removing the size selection. And we've removed the small fragment elimination step. And we can do that because we're starting with fresh blood. Just as a side note, the library prep for ONT doesn't need to be high quality, high molecular weight DNA. Obviously, it helps to have high quality DNA with the first steps. So at this stage, you'll have extracted your genomic DNA from whole blood. You'll have quantified it, and you will have shared it with the megariptor. You'll have 3 micrograms of megariptor shared DNA, and then you go on in using the classic ligation chemistry. So the library starts with N repair and DA tailing, and then we perform the usual ethanol-based cleanup step, and then a qubit quantification for the end prep library. Then we proceed with the adapter ligation. We do 2 careful wash with the long fragment buffer, and we proceed to the final quantification. So you should expect to recover between 1200 and 1800 nanogram of adapter ligated library in a volume of 96 microliter. And that's enough for you to load over 3 Promethean flow cells at the same time, so simultaneous. You can refer to the Promethean flow cell loading part of the 24-hour whole genome protocol, and I'll share the QR code later for more info on the preparation of the sequencing library. But as you can imagine, you simply need to do 3 times the standard volume. So no washes are needed. The library is sequenced for 13 to 16 hours to reach 30x coverage. If you want to go to 40x coverage, it should be approximately between 18 and 26 hours. And then for the analysis, you will then perform hack, live-based calling, alignment, and BAM concatenation on the instrument. Again, Jawar is going to explain all of that. The downstream analysis is done on the instrument at the end of the sequencing run using the human variation workflow on Epitome desktop. And the tertiary analysis is outsourced to one of our third-party company, again, like Genix or Fabric Genomics. A very quick last slide on the Promethean 24. So in our case, we use 3 flow cells per sample, but I just wanted to put a little bit more information about the instrument itself. So you can run, if you do not require the 24-hour. So if you do one sample per flow cell, you can achieve 30x whole genome. If you run the instrument at full capacity, you can run and achieve 48 human whole genome per week at 30x. That's just short of 2,500 genome per year. And on that note, I'm now going to hand over to Jawar.
Thank you, Cora, for the introduction on the rapid WGS, the 24-hour WGS protocol. I'm going to be talking about how to set up the experiment, which basically involves how to set up sequencing, how to do the secondary analysis, and how to do the interpretation part of it. So my presentation is split roughly into 3 parts, lots of screenshots and lots of detail, but I'm not going to be talking through most of the detail because they're mostly self-explanatory, but let's get started. So if you look at the protocol, this chart here divides it into the different sections that you have to do during the experiment, the 24-hour WGS experiment. So we are starting at this point where we are starting to set up the run, and then we'll do the sequencing, and then there'll be the other sections, as I mentioned, on secondary analysis and reporting, and tertiary analysis and reporting. So briefly, for those who are in a hurry, what does this step involve? It basically involves setting the basic parameters for sequencing, which includes getting the right reference genome sequence for your experiment, configuring MinKNOW, which is our instrument control software, and then parameters that you need to change to enable multiple flow cells to be run for the same sample. So there are some specific details I'll be touching upon. And finally, setting out the output options so that the next steps that follow the sequencing are not a problem. So jumping right into it, configuring an experiment in MinKNOW, you have to obviously choose the 3 flow cell positions you need to run and enter an experiment name. Now the critical thing here is that you need to give the same sample ID for all the flow cells that are going to be running in this experiment. This is critical because this ensures that all the data is written under a single directory tree, which makes it easier for the analysis to pick up these files at a later step. So if anything you need to take away from this is choose an identical sample ID for your experiment. There are bits and pieces you need to set up in MinKNOW when you're setting up an experiment, primarily the ligation kit, what sort of base calling mode we have, and we recommend running hack base calling, followed by alignment, and ensure that the modified bases are turned on. Add the reference genome FASTA file that I mentioned, get the right sequence reference, and then add it to the thing. Set the runtime to 24 hours and leave all the other parameters as default. Importantly, select one BAM output at the end of the run. So you have 3 flow cells running. Each flow cell needs to be set in such a way that it writes out a single BAM file at the end. So you have multiple options there. Click on BAM and then choose end of run and then click save. So that ensures that the sequencing will happen for whatever time necessary, which I will touch on in the next slide. And then the BAM file is written at the end of that experiment per flow cell, per sample. How long should we sequence for? This keeps coming up. So sequencing time obviously is dependent on a couple of factors. One of them is the number of available pores on the flow cell, and then the read length or the N50 of your library. So the graph on the right hand side basically shows, depending on the kind of pore, number of pores you have, the amount of time you're going to require for sequencing. So in the experiments we've done while setting up this protocol, we discovered that if you had about 5,000 pores, it takes 3 flow cells taken together, take about 16 hours to hit 30x coverage. If you have more pores, then obviously it takes much lesser time, but it's a good idea to keep that in mind when you set up the experiment so you know when you can stop the experiment. And this is a typical example. We ran 3 flow cells, each with close to 7,000, 8,000 pores, and then you can see the output over a period of 12 to 14 hours, I think, is when we ran this, and we got a total of 162 gigabases of data of the 3 flow cells, which is 52x if you assume 3 GB for a human genome reference. And that just shows you how the pores perform as as a parameter of time, and you can see the number of available pores going down. The interesting thing to note here is that at the end of this experiment, there is still a lot of pore capacity left, and that you can probably wash and reuse the flow cells for different purposes later on. But for this particular experiment, 12 to 14 hours gives you at least 30x coverage across the 3 flow cells. So that's your sequencing. Now, hopefully you've done this and you've pressed start and you've gone away for a good night's sleep, or spent the rest of your day doing other things. And you come back and you notice that the data has now been generated. It's more than 30x. It's been about 12 to 14 hours since you started sequencing. You can now stop the sequencing and then basically go to the next step. So that gets us to the next box, which is the secondary analysis and reporting, which is 3 to 4 hours. One thing to remember here is, because the machine's busy doing sequencing and the compute that's attached to the sequencer is busy handling the sequencing software, we do not recommend sequencing and performing data analysis simultaneously on a device. So let the sequencing finish and then start the secondary analysis part of the process for the 24-hour WGS. For this, we have our own platform, bioinformatics platform called Epitome. We recommend using human variation workflow, which is WF human variation workflow version 252 or higher. I would basically say the latest versions are the better ones. So you have 272 or 273 are available. So install that on your Epitome desktop application version. The workflow is an orchestration engine. So you basically give it a few bits and pieces of data and it goes and does all the variant calling, the alignment, and produce various reports and write out a set of VCF files at the very end. There are some hardware and software requirements for running WF human variation. We generally recommend 32 CPUs per job and with 128 gigabytes of RAM, and you need about half a terabyte of space to be able to run this. Given the amount of resources, it will take approximately 3 hours per sample to run on a PromethION 24. We do not support ARM processors, and if you're using a Mac, the M-type chips are not supported. But apart from that, it's just quite simple to install and run, and there are a few boxes you need to fill in. And here we show you how to set your working directory to a particular location so the workflow analysis does not crash. This is assuming you're running the analysis on the same compute that the sequencer is running on. Again, briefly, for those in a hurry, there are a few things you need to do. Obviously, there are some main options that you need to turn on. Basically, turn on SNPs, SVs, CNVs, STRs, provide the necessary inputs, provide some additional options, and then optionally provide partner integration. So we have partnered with Fabric or GeneYX or Genix. They are interpretation partners. You can choose if you want to write outputs in that particular format, which support either Fabric or Genix. Again, the basics of setting up human variation, you just launch the workflow, fill in a lot of boxes, ensure that you've turned on SNP, SV, CNV, STR, and modifications in the workflow options that are provided to you. And as you fill up these options, you can see that the boxes will start to turn green, and that everything that you needed to provide here is available for the analysis to happen. Now, do you have to select all variant types? Probably not, because some tertiary companies support some variant types and some don't, but I think it's a good idea to support write out all variant types, because it forms a complete record for the experiment that you've conducted. Now, remember the sample ID I spoke to you about before is come back again. So in this time, when you set up your experiment in human variation, you need to provide the same sample ID used in MinKNOW, so that the experiment knows that the data came from 3 different flow cells and it can actually put them together. And that is really, really critical. If you don't do that, you'll probably pick up the contents of one of the flow cells out of the 3 and you won't get enough coverage. We also recommend you turn on phasing. We use WhatsApp to phase variants. This basically enables us to use the longer reads and the patterns of variation amongst the long reads to be able to split variants across the 2 haplotypes. So you can get haplotype 1 and haplotype 2 ideally one of these haplotypes comes from either parent. So you can start differentiating variants that occur on one chromosome versus variants that occur on the other copy of the chromosome in a human. Partner integration, I referred to that briefly earlier. So if you have a licensing or agreement with Genix or Fabric, you can choose partner integration for either of them, and depending on which option you choose here, you'll get different output files at the end. For Genix, you'll get 2 VCF files, one that contains the small variants and another VCF file that contains all the other variant types. For Fabric, you'll get a single VCF file that merge all the variants that are available from the output of the human variation workflow. So here are some basics here. So again, it's just a recap of what we spoke before. So set all your main options, set your sample ID to be the same sample ID as you set up the experiment with, and then set up additional options around phasing and press the launch workflow button, and then you can then go away for a couple of hours while the job runs. Now, if you're like me, then you probably don't like using desktop or Windows and point and click. It is also possible to use the command line interface to run human variation workflow, and I've just provided an example of the kind of command line you would need to create in order to run this job. I won't go too much into this, but it is possible, and reach out to us and we can always help you set this up at your end. At the end of the human variation analysis, you get a lot of reports. You get one report for every process. So you get an alignment report, you get a SNP report, you get an STR, and CNV and SV report. So it's a good idea to just glance through them to see whether the data looks okay and there isn't something seriously wrong. For example, in alignment, you want to see that most of the data you've collected has mapped to the human genome, and it's not mapped, and it's not a particularly low number, because that would indicate contamination. Like I said before, with partner integration, the merged VCF files, depending on whether you chose Genix or Fabric, are available to you in a separate directory. And that completes secondary analysis, and we come to the final part of my presentation here, which is going to be on tertiary analysis. And that's the last box here. And we've only said 2 hours because this is the kind of time you would need to upload all the files to the tertiary analysis partner. This is not the time that's going to be taken for necessarily interpreting the genome. What do I mean by tertiary analysis? Well, in our world, tertiary analysis or variant interpretation is the analysis and identification of variants associated with a specific trait. It could be a disease, particular phenotype, or a particular population. In this case, of course, we're talking about 24-hour WGS, so this is about identification of variants associated with a specific disease or a specific phenotype representation. One thing to note is Oxford Nanopore does not offer tertiary analysis solutions themselves, but we instead have partnerships in place with companies like Genix and Fabric to create output files that are compatible with those providers. Another thing to add is Genix and Fabric are not the only partners we work with. Their other compatible products are available for Oxford Nanopore data, which are listed here. I think there's one more listed here, which is Safator, Varsome. So what's a compatible partner? We spend some time and effort to certify that the third-party providers that we work with are compatible with the output from our secondary analysis pipelines. We do not look into licensing, we do not look into pricing agreements, and because these are beyond the remit of Oxford Nanopore. So if you're interested in using one of our compatible partners, our recommendation is to reach out to them and negotiate directly around the pricing and licensing issues if you choose to use one of our compatible solutions. And finally, and here's hoping all your experiments go well and the 24-hour WGS is successful. Oxford Nanopore will support you. Our technical support team is available to support you, particularly at every step till this generation of the VCF files. So that's got to do with, setting up the sequencing, extraction, library prep, secondary analysis, and then, and then if you have any problems, you can always get in touch with us. Any queries relative to tertiary analysis should be addressed directly to the regional technical support for your tertiary analysis provider. And, and I will leave you at that point and basically say good luck with the 24-hour WGS and hope you're successful. And it's been a pleasure talking with you. Thank you.
Thank you very much, Jawara. So I'm just going to go quickly over a few resources that you guys might need to look into the protocol more. So in terms of consumable and equipment that you need, in consumable, as we discussed, you're going to need the sequencing kit, the flow cell for the ProMethane, the pure gene blood kit for the DNA extraction, then a couple of third-party reagent from NEB, megaruptor shaving kit, and then adjuvant genomic DNA analysis kit and a qubit kit for the DNA quantification and quality checking. In terms of equipment, you'll need a ProMethane 24 sequencer, the instrument for the megaruptor, the femtol pulse from Agilent, and a qubit fluorometer. You might want to have a look at the workflow overview, and we put the QR code for you here. It's just a very high-level summary of what we've presented today. And then for people who really want to look into the protocol, it's available on our website, and the QR code is also available here. So very quickly, as a summary, the 24-hour whole genome sequencing application with Oxford Nanopore offers the most comprehensive genome, covering most of the variant type with the longest reads available for our audience. The 24-hour genome protocol is the fastest whole genome sequencing solution available that also includes methylation. Oxford Nanopore human whole genome sequencing shows a lot of future potential to push diagnostic yield higher as it includes structural variant and methylation, and as those tools improve in the future. Very importantly, there's no penalty for sequencing the proband only. Unlike other solution, you can just do the proband as it arrives. And an Oxford Nanopore sequencing device really offers the smallest footprint, which can be really important for some lab where room is really a limiting factor. And on that note, I will hand the mic over to Mark, again, who is a laboratory specialist at Erasmus Medical Center. Over to you, Mark.
All right, thank you so much. So my name is Mark Drost. I work as a clinical laboratory geneticist at the Erasmus Medical Center in Rotterdam, which is in Holland. And this is my disclosure slide. And the Erasmus Medical Center is home to the largest neonatal and pediatric intensive care unit in our country. And we admit about 2,300 patients every single year. So in about 10% of those cases, a clinical geneticist is consulted, which means that there is a genetic disorder suspected. And in about 10% of those, so about 25 cases per year, a clinical geneticist will ask for rapid genetic testing in our center. And it is not that long ago, about a year or so ago, where our rapid genetic testing flow would take 21 days. Our testing flow was mainly short-read sequencing based, and then depending on the case, those short-read genomes would be supplemented with additional testing, such as, for example, for DMPK or for SMA. Also, short-read genome sequencing is built for throughput and for low cost, but it's not so much built for speed. And even for us, as a center, we have outsourced our sequencing. And as you might imagine, especially for these critically ill children, 21 days is way too long. Parents are very worried. Doctors want to make life or death decisions in the first few days after hospital admittance. 21 days is still way too much. So can we improve this? And we thought, yes, we probably can by replacing our short-read sequencing flow by ultra-rapid nanopore sequencing. The main advantages being that you work sample per sample, which makes you fast. Also, sequencing is done on the machine and it's much faster, also with base calling done on the fly. We actually have the machine in-house, so we do not outsource. And because long-read genomes provide a much more comprehensive overview of our genome, we probably need a lot less additional tests. So we're probably also making genetic testing much more efficient for these children. So this is our testing flow that we use. So our doctor is able to request ultra-rapid genetic testing to our patient management system, and when they do, a technician will start on sample prep almost immediately. The technician will take approximately 4 hours. So after 4 hours, our promethylon is sequencing. We do all patients in a trio fashion, so we use index plus blood of both parents. We do sequencing and base calling on the fly. And I know Oxford Nanopore uses 3 flow cells per DNA sample for their ultra-rapid flow, but we actually use 2, so we use 6 flow cells per trio. We do all of our variant calling on our tower, and it is actually quite comprehensive. We do SNV, CNV, and SV calling, and we also call regions of homozygosity. We score for contamination. We have an SMA caller built in. We look at DNA methylation signatures, and we have SDR calling in progress. We annotate and prioritize our variant in a tertiary analysis package, and then afterwards data gets analyzed by a technician and by a laboratory geneticist like me, after which I write a diagnostic letter to a doctor. So this is our end-to-end flow that we have incorporated, but before we did, we have done a year-long clinical utility study for ultra-rapid nanopore sequencing. And for this utility study, we have enrolled 26 critically ill patients, and for every single patient, we have asked the involved clinical geneticist to ask for our standard of care, which was still short-read sequencing based a year ago, and we run ultra-rapid nanopore sequencing in parallel. So basically we have 2 arms, a diagnostic short-read arm and a research-based ultra-rapid arm. And once those analyses are finished, we compare turnaround time, diagnostic yield, and effect on clinical decision-making for both techniques. And then these are our results. We have enrolled 26 critically ill patients with a median age of 2 months, so often they're young, but we've also included 3 adults already. And then this is probably the most important figure in my presentation. So here I show you average turnaround time for these 26 cases using standard of care, so short-read genome on the left, and also nanopore sequencing on the right. And I hope this figure convinces you that using nanopore sequencing, we were able to reduce our turnaround time by 2 weeks. So simply by changing platform, we gain 2 weeks, which especially in this ultra-rapid critical setting is essential. We are so fast that actually genetics are now part of clinical decision-making at the ICU. And also to stress, this includes the weekend. These are actually calendar days, they are not working days. But then we're pretty fast, right? Are we still being accurate? And we think we are, because for both arms, so both for our short-read genomes as well for our nanopore long-read genomes, we have compared diagnostic yields, so the number of diagnoses that we've made, and we have actually solved 11 out of 26 cases in both arms. So we are both being fast, but we are also being pretty accurate. But of note, our standard of care, also our short-read flow, is often a combination of short-read sequencing plus additional testing, meaning either STR testing for DMPK, methylation testing for Prader-Willi. For nanopore sequencing, we always do only one. So we have the same diagnostic yield, but we use less tests. So we're not only making genetics faster, but we're also making genetics more efficient. And then, last but not least, we have also looked at decisions on clinical management, and we have actually solved, for the 11 solved cases, for 7 of those we have direct impact on clinical management. For example, because our genetic testing result led to a medication switch or a termination of treatment. So in the graph that I'm showing you here, the second graph from the left, that is the average turnaround time for our 26 cases. In our next slide, I will actually show you turnaround time for every single case individually. On the y-axis, you see every single case numbered, and on the x-axis, you see our turnaround time in days. I have 3 colors in the beginning of every single patient. Those represent our ultra-rapid nanopore sequencing flow, and then the gray bar represents the turnaround time for our short-read genomes. And you can hopefully appreciate that for every single case, our ultra-rapid nanopore sequencing flow is a lot faster than our short-read flow. And also these cases are ranked by turnaround time, the fastest one being on the top, so the fastest using nanopore sequencing on the top and the slowest on the bottom. And then the yellow circles that you see for every single case, they represent a diagnosis. So you will hopefully appreciate that all solved cases are in the top of the graph, meaning that for solved cases, on average, we are really fast. Actually, for solved cases, we have a turnaround time of 4.2 days. So we have an average turnaround time of 5.2, but for the solved, we're even faster. Mainly because for the negative ones, we do multiple analyses. Then the great thing about nanopore sequencing is that the test is so comprehensive, which I will illustrate with these 3 examples. So first of all, we were able to directly confirm an autosomal recessive disease in a singleton case, so in the absence of parental DNA, which is exemplified by the case here on the left. We were able to directly resolve both haplotypes and confirm autosomal recessive inheritance. Then for a second case, we were able to diagnose an mtDNA disorder, which is also accurately picked up using nanopore sequencing. And in one specific case, we were able to identify a CSD7 or ICJ8 charged DNA methylation signature profile in our nanopore sequencing data, which then made us able to directly investigate the CSD7 locus and identify a pathogenic CSD7 variant in that patient. So now I will show you a few examples of some cases that we've worked with. So the first case is a 10-month-old boy. He presented with seizures and with gray hair and skin lesions, and he also had white matter lesions on the MRI. So these symptoms combined, for our neurologist, he had a differential diagnosis of Griscelli syndrome. For this case, our clinical geneticist was consulted at day 1. She asked for standard of care using short-read genomes and ultra-rapid nanopore sequencing in parallel. We were able to isolate DNA on day 2, and then already on day 3 we identified a homozygous pathogenic variant in the RAP27A gene, confirming Griscelli syndrome and making this kid eligible for stem cell transplantation. As we were still in the research phase, our ultra-rapid nanopore sequencing results were still confirmed, in this case using SAR sequencing, which we did on day 11, so 8 days later. And actually our standard of care results came in only on day 22. So in this case, using nanopore sequencing, we have gained 19 days of speed just by using nanopore sequencing alone. Then in our second case, we have a 1-week-old boy. He had a very severe case of epilepsy and decreased consciousness. Clinical geneticist was consulted on day 1. We had a weekend in between, so it took us a bit more time to isolate DNA, which we did on day 5. And then 2 days after that, we identified a pathogenic de novo variant in the KCNQ2 gene, explaining his condition and confirming a diagnosis. And the interesting thing for KCNQ2 mutations is that these patients actually are eligible for different therapy types. So the doctor switched therapy for this kid, and he was given different types of medication as a consequence of the KCNQ2 diagnosis. But unfortunately for this kid, these results did not lead to improvement, and unfortunately he died on day 21. But needless to say, in this case, the standard of care results came in only on day 15. So we were able to switch therapy 8 days earlier as a consequence of nanopore sequencing. And then in the first 2 cases that I showed you, they mainly show you increased speed. But in the first case, I'd like to show you that nanopore sequencing is a much more comprehensive test and is also therefore much more efficient. So case 3 is a newborn boy. He had a respiratory insufficiency and a growth retardation, and he presented with hypertonia. So the involved clinician in this case, she asked for short-read genome for metabolic disorders, for muscle disorders, and also a larger OMIM panel. And she also asked for SMN1 and DMPK testing in parallel. And for this case, she did not ask for Prader-Willi testing, because for these small infants, we often have small blood volumes and therefore a small amount of DNA. And for that reason, she chose not to test it, also because Prader-Willi was not too high in her differential diagnosis. But then nanopore sequencing clearly showed a 15q11 deletion in the Prader-Willi syndrome region. And because on nanopore data you can also look at methylation, we were able to directly show that in this case the paternal allele was deleted on that locus, which fits with the diagnosis of Prader-Willi syndrome. So even though the doctor in this case did not ask for Prader-Willi testing in standard of care, we were able to directly diagnose Prader-Willi on our nanopore data because it's just such a comprehensive test. Alright, which then brings me to my conclusions. So I hope that I've convinced you that for us as a center, by switching from short-read genomes to nanopore sequencing, we have reduced our turnaround time for these critically ill kids from 21 to 5 days, and actually even 4 days in case the case is solved. Our diagnostic yield is similar to short-read genomes, so to our regular diagnostic testing, but we need a lot less tests. So we're not only making genetics faster, we're also making it more efficient. And in those cases that we solve, we have an impact on clinical management in 2/3. So it's actually, the impact on clinical management is quite big. And then, which then brings me to my perspective. So first of all, as a center, we are constantly trying to speed up. So we now have a turnaround time of 4 to 5 days, but we actually like to move to 3 to 4 days continuously. And one of our major bottlenecks in our center, at least, is that it still takes us quite a bit of time to isolate DNA. We're a big center, so often it takes a lot of time to transport blood from our ICU to our department, which may seem trivial, but it's just not that easy in our center. Also, we see this as a step towards a broader implementation of nanopore sequencing in general, but also ultra-rapid applications in a more specific way, specifically for adults at the ICU. But also, we would like to use nanopore sequencing after aberrant newborn screening. We're also doing cost efficiency studies. So for us as a lab, nanopore sequencing is quite expensive. We use 6 flow cells per trio, but you could imagine that for the Erasmus Medical Center, in a broader sense, this is actually very cost effective because we take so little time to influence clinical decision making, and probably our results in the end will lead to shorter ICU stays. And then last but not least, we have also conducted interview studies. So we have interviewed healthcare providers as well as parents of ICU patients, and they have considered nanopore sequencing a valuable enhancement to ICU care, and they have also stated that nanopore sequencing helps to reduce this uncertainty period that they face when their child is at the ICU. Alright, so with that I'd like to end. I'd like to thank all the people involved, but in particular the ultra-rapid nanopore team, nanopore sequencing team, and I'd like to thank you for your attention, and I'd be happy to take questions.
I'm afraid that's all the time we have for today. So thank you again, Mark, Jawar, Eli, and Vanisha, and everybody on the audience for joining us. I just wanted to remind everybody that our product of our research is only. You can find more information on the 24-hour whole genome sequencing workflow using the QR code or to download the link that's on your screen. This knowledge exchange, along with all of our previous broadcasts, are available for you to watch on demand. So please visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinar and knowledge exchange on the Oxford Nanopore website.